Okay, so we're back in part 2, and essentially I'm just breaking this lecture video up into small sections so that they record and upload without any trouble, without any technical issues. So we're back with part 2, and we're talking about this sodium-potassium pump. Okay, so the idea behind the sodium-potassium pump is we talked about how particles will move in according to concentration gradient, right? And we talked about when you have solutes on the inside, when we were talking about our dialysis tubing example, you would have solutes on the inside in a great concentration, and then on the outside you would have water or plasma or something of that nature here. When this area is more dilute, this area here is more dilute than this area inside. Now, these solutes, specifically what we're talking about is sodium and potassium now, are not readily able to cross the membrane as they need to, necessarily, passively. What I'm saying is they can't diffuse. So if they're not diffusing, by increasing solutes in here, we can pull water in, right? Because then water will osmose down its concentration gradient, and it will swell the cell. It will swell the cell. And we're not talking about doing it to the extent that the cell will lyse or break. We don't want the cell to break, but what it does do is it allows us some kind of a mechanism of control over cell volume, okay? That is the key here. We are controlling cell volume, and then you'll notice that these little ions is what they're called, have this charge on them. So by controlling the number of ions and the number inside and the number of ions outside, we kind of can control the charge on this cell, whether it's more positive or more negative in relation to the outside of the cell. And these two things, cell volume and charge, are something that we can use, all right? So we have this mechanism, this cellular mechanism that we're going to talk about, which is called the sodium-potassium pump. And the sodium-potassium pump allows us to sort of control those two factors of the cell. It helps us, there we go, sodium-potassium pump expels unwanted ions, stockpiles the needed ones, and helps us maintain cell volume, right? So when they're talking about expelling and stockpiling of ions, what they're talking about here is charge. It allows us to change the charge in the cell, which will become important later when we're talking about action potential in muscles and neurons, okay? So this is one of the mechanisms we use, again, to control cell volume and charge. And it is a form of active transport. Let's get that clear. It's active transport. And why is it active transport? The answer here lies in our second point here, and it's because it is using ATP. This reaction, this transport, sodium-potassium pump, cannot happen unless we utilize ATP. Now again, if we want to increase cell volume, we put more sodium inside the cell, and if we want to decrease cell volume, we put less sodium inside the cell, or actually what we do is we increase the expelling of more sodium, so we put more sodium outside the cell. And the idea here, the thing that you want to keep in mind, is this principle that we always, I'll say it a few times over the course of this class, but the idea here is that water always follows salt, okay? Water always follows, oops, I spelled my salt wrong there. Water always follows salt. And if you remember that, it's kind of a little mnemonic for remembering what's happening here and why it's happening. So again, if we want to increase the cell volume, we need to put more water in that cell. So we're going to move sodium into the cell. And then if we want to decrease cell volume, we want to move water out of the cell, so we're going to push sodium out of the cell, and water will follow and the cell will shrink, okay? So that is the idea. So let's move forward and we're going to take a look at how we do that. And you know I love my diagrams, so we're going to start with this diagram here, okay? Now we talked about, let's zoom in here and take a look at the energy being used. Here it is right here. We know that this is active transport because of this guy right here. We utilize this ATP to make this whole process happen. And you remember I said that ATP stands for, oh, my pen is kind of thick here. Let's see if I can make it a little smaller for our purposes. All right, let's try that. ATP stands for adenosine triphosphate, okay? Because there are 3 phosphates. Adenosine triphosphate. And we talked about this third phosphate as being sort of a high energy molecule, okay? High energy. And what I said is that when the cell needs to utilize ATP, what it does is it takes this here, it cleaves off, tears off one of these phosphates, and that energy is released and used by the cell for some purpose. And then we're left with ADP. We're actually left with ADP and this free-floating phosphate out here, this phosphate ion, or as it's noted here, PI. And these are sort of floating around the cell. We've got ADP and PIs floating around the cell until they're taken into the mitochondria and they're sort of shoved back together through a process and they become ATP again, and that's how the mitochondria got the name of being dubbed as the powerhouse of our cell, okay? So we talked about that. That's a little refresher. So now let's back out and take a look here at what's going on. So for whatever reason in this example, we want to bring water into the cell. We're looking, the goal here in this example is to bring water in. We want to bring water in. I'm going to write that here so we can remember. Goal, or we'll say the goal is to increase water in the cell. Okay? We're going to increase water in the cell. My sloppy writing on my iPad here, but I think you can follow. So here's how this is going to happen. We've got this transmembrane protein, this integral protein that is going through the plasma membrane. It's available on this side and it's available on this side. And we talked about what it might need, why it might need to be configured this way so it can communicate with the inside and the outside and perform some type of function for this cell. And in this case, the cell is readily open to the extracellular environment at the start. And it's got a place in here, a designated configuration for 3 sodiums to load up in here. So it sits here pretty much ready all the time in this configuration. It's sitting here loaded with 3 sodium. Now nothing happens to it until it reacts with the energy taken from this ATP. So ATP comes along and it reacts with some region of this protein which causes the release of this high energy phosphate. Actually, the phosphate's not high energy, but the bond between the ADP and this third phosphate is a high energy bond. So when we break this phosphate off, we release that energy, and that releasing of energy then causes this protein to change shape. So maybe it went from this knotted up configuration into like a loose whirly configuration or something like that, just to give an example. But after it changes shape, what happens is it releases these sodium into the extracellular environment, Now I'm going to just correct myself here because this picture is backwards from the way I'm used to teaching it. So we're actually looking at the goal is to move water out of the cell. move out of the cell in this particular picture. So then once we've cleaved this and this high energy is utilized to change the shape and we release the sodium, now this new configuration, instead of having 3 sodium pockets, it has 2 potassium pockets. You see that? And they load up inside of there. Now the binding of these potassium to this wall creates another configuration change in this protein, and it opens up to the inside and allows the release of these potassium into the cell. Okay? Into the cell. Now, if we've shoved 3 sodium out and 2 potassium in, let's assume this is a 1-for-1 exchange, we've got an uneven number now, right? We've got 2 ions on the inside and 3 on the outside. So what we've done is we've increased solute concentration outside the cell. Now what's going to happen? We're going to have some osmosis occurring. So sodium went out, or solute concentration went out. We've created a situation where we have an area. Let me erase some of this garbage so that we can see what I'm talking about now. We have this area of high concentration, 3 on the outside, to low concentration, 2 on the inside. And water is going to diffuse, or excuse me, it's going to osmose across in order to sort of keep this dilution the same. It wants, what did we say? It wants a isotonic situation to occur. So water's going to move, the cell is going to shrink a little bit, but here's what happens. We have these leaky potassium channels, potassium channels. So slowly over time, potassium will leak out. And that will trigger the cell to start reacting again and saying, hey, we need some of this potassium back in the cell. So in order to do that, it's going to trigger this whole entire process again, and it's going to ask for sodium to leave the cell in order to bring back potassium in. And it's a cycle. And if you think about it as being like the what's a good example? For you mechanically inclined people out there, your air conditioner compressor on your automobile. So when you turn the AC on in your car, this compressor will run until your car gets to temperature, and maybe then it will kick off, and then it will turn on again when your car starts to warm, and then it will kick off, and it just goes in a cycle. So this happens. We bring sodium out. We bring potassium in, the cell is happy for a little bit, but then some of that potassium leaks across, and so then we say, okay, we're going to kick some more sodium out, and we're going to bring some potassium in. Now we're happy, then it leaks out. We do it again and again and again, and it's a cycle. Now, you might be asking where we get this sodium, and realistically, this is not the only transmembrane protein that's at play in our cell, but this is one that we understand, so it's a great one to articulate sort of how proteins can work, and this is the one that we like to study. But there's other proteins here and here and here on the cell that are doing different functions, and at some point there's sodium that's coming in as well. There's sodium that's coming in, there's potassium that's going out, but the idea is that this potassium is leaking across faster than the sodium, and so we've got to pump some of this sodium across in order to bring potassium back in and keep it balanced. So it's kind of a messy thing, But know the steps to the sodium potassium pump. You will be tested on it. Know that it is an active transport mechanism. Why? Because it uses ATP. And again, the ATP is cleaved into ADP and a free phosphate ion, and that breaking of that high energy bond allows this protein to utilize that energy, changing shape and driving this whole process. Now, if ATP were not present, we wouldn't be able to do this process. The sodium potassium pump would shut down if the cell were not producing enough ATP. When might the cell might not be producing ATP? Well, we talked about that in class, and so I'll just pop it up here as a reminder. If we had an anaerobic environment where we were not getting enough oxygen and therefore our mitochondria production of ATP is down, then we might have a low ATP situation, which might affect the functioning of this sodium potassium pump. Now, what else could affect the function of this sodium potassium pump? Since we're here, we'll just talk about it briefly. If we had some kind of electrolyte problem, for instance, if we had low sodium levels, we don't see that as much, but what we do tend to see is some kind of pathology that has to do with potassium. So sometimes patients will be told, listen, your potassium is low. It's going to trigger all kinds of problems in the maintaining cell volume and the maintaining electrical gradients across cell, and your cells are not going to be able to function properly, so that patient will be supplemented with potassium. And then things will start to kind of kick in and work the way that they're supposed to work. So there's some ideas. That's the sodium potassium pump for you. And then we're going to go ahead and pause the video here, and then we'll open up with another quick section in a moment.